Okay, I will start slowly. So welcome everyone to today's Monash Advanced Microscopy webinar. And first, I wish to acknowledge the country. Monash University acknowledges the people of the Kulin Nation on whose land our university is located, and pay our respects to elders past, present, and emerging. It is my great pleasure to introduce you today to Professor Masaru Ishii. Professor Masaru Ishii graduated from the Osaka University Medical School and then worked as a physician specialized in rheumatology and internal medicine. He studied at the National Institutes of Health as a Human Frontier Science Program long-term fellow and as a laboratory chief in Osaka University Immunology Frontier Research Center. He then was appointed as a professor and chairman of the Department of Immunology and Cell Biology, Graduate School of Medicine, Osaka University. Professor Masaru Ishii has authored over 150 publications and has received several prestigious awards. I will only mention some of them. These are the Young Investigator Award, the Japanese Medical Association Award, the JSPS Prize, the JSBMR Ogata Prize, the Japanese Society for Bone and Mineral Research Prize, Setsuru Abashi Prize, and the Osaka Science Prize. He serves also as a director of International Federation of Musculoskeletal Research Societies, is the vice president of the Japanese Society of Bone and Mineral Research, and the director for the Japanese Society of Inflammation and Regeneration. Professor Masaru Ishii is also the director of Nikon Imaging Center at Osaka, a part of a worldwide consortium of biomedical imaging research. Professor Masaru Ishii, it's our great pleasure to have you here today and to be able to listen to your talk. The stage is yours. Thank you very much.
Thank you very much, Jennifer. Kind introduction. So, first of all, I call-san, the organizer of this series seminar, for giving me this wonderful opportunity. So, I am Masaru Ishii, a professor of immunology and cell biology at Osaka University School of Medicine, and have been studying on immunology, especially focusing on immune cellular dynamics by using intravital imaging technology. So, because Monash and Osaka are currently trying to make close collaboration in any fields, I would be most happy if I can collaborate with you all in live image technology in your hand. Okay, so anyway, let's start the presentation. So let me share the screen.
Yes.
Okay. Can you see the screen? No?
Yes, there it is. Yes, it looks good.
Okay. So, and also, okay. So, because my presentation contains lots of movies, so during the presentation, I stopped my video. It's okay. So, probably, yeah. Okay. So, yeah, it's it can reduce so subtitle transfer. Okay. Anyway, so yeah, the title of my presentation, today's talk is Intervital multiphoton optical imaging dissecting immune cell dynamics in vivo. Okay, so yeah, this is the agenda. Today's talk, and in the first part, I will explain, especially focusing on the imaging of live bone tissues. So as you know, there are several kinds of live imaging. Live cell imaging is visualizing live cellular behavior in vitro culture. Live tissue are also cultured and visualized, although These techniques are based on the removal of cells of tissue to be interested from living body. On the other hand, Intravital imaging in the hand enables us to visualize real cellular movements in completely intact conditions. With this technique, we have first revealed the system for visualizing inside the bone. So as you can imagine, bone is the hardest tissue in the body, and it is difficult to pass through the excited lasers. Anyway, so we finally succeeded, and moreover, we succeeded in imaging cellular dynamics in these tissues organs intravitally. So this is an example for visualizing peripheral lymph nodes intravitally. So mice are anesthetized by isoflurane, and systemic conditions are monitored, and so they are kept alive and healthy. So I'm teaching immunology for medical students, and this is a typical snapshot from an immunology textbook, where the immune cell dynamics are explained in plausible cartoons, but we are not sure if these cartoons are really true or not. And by using our imaging technology, we could grasp the real cellular behaviors like these. Oh, sorry, this is Japanese. Sorry. So we know antigen presentation is much more dynamic than we expect in cartoons. And because I am originally a rheumatologist, seeing patients for rheumatoid arthritis, so I have been interested in inflammatory bone destruction by osteoclasts. So osteoclasts are especially differentiated macrophage, having a unique capacity to destroy bones. There have been lots of studies on osteoclasts, although most of them were based on the static histology or in vitro culture experiments, and their in vivo mode of function, especially its dynamic nature, has remained unknown. So that was why I tried to analyze them in vivo. So this is an outline of the method. So we are utilizing multiphoton microscopy, which enables us to visualize deep inside the live tissue with less photo damages. And in the case of bones, by using conventional fluorescent microscopy with visible X-ray lasers, lasers can only be passed through around 30 to 50 micron depths, and then we cannot reach inside. Infrared lasers for two-photon excitation can be passed up to several hundred microns deeper inside the bone tissues, so then reaching to a bone cavity inside. And by means of this technique, we have first revealed the mode of migration of osteoid precursor macrophages. When I started study around 50 years ago, it was still unknown whether osteoclast precursor macrophages were originated from local bone areas or they supplied from blood circulation. With this imaging technology, we clearly demonstrate that osteoclast precursors are recruited from blood circulation, and it was regulated by a kind of lipid mediator named sphingosine phosphate. So let's focus on the final stage of osteoclasts. Here, those are actually destroying bones on the bone surfaces. And for visualizing mature osteoclasts in vivo, we made a reporter system by conjugating GFP with a subunit of V-type proton ATPases. That is an essential machinery for secreting acid and resolving bonds. So here's the result. So there are bone destroying mature osteoclasts in vivo. And please look at these cells carefully. And then we can identify two types of cells. One is static, the other is moving. And so, yeah, here's a static one. So in static ones, the GFP fuse the proton pump, the green signals, distributed along the membrane. It's a single membrane. So it's a firmly attached to the bone substance. And GFP fused proton pump are accumulated on the single membrane, meaning that these are now secret acid towards the bones. Then we name these osteoclasts as bone-resorbing R-type osteoclasts. On the other hand, in moving ones, proton pump are distributed not to the membrane, but in cytoplasm. So, yeah, because they cannot secrete acid to outside at this moment, then we call these as non-resorptive osteoclasts, N-type osteoclasts. So now we can discriminate which is functional and which is not. So in another study, we try to analyze actual event of bone destruction. So in order to directly visualize bone resolving function of osteoclasts, here we generate a chemical fluorescent probe for detecting a proton secretion at local bone surfaces. So this is a pH sensing chemical probe here. So it is area, so they can sense a proton or pH. And here, these two structures can firmly attach to the bone. These are bisphosphonates. So then this is a pH sensing chemical probe attaching to bones, and green signals, clear signals can be turned on if the local pH is fewer than 5. So yeah, this was done in a collaboration with chemical biology in Osaka University. So here's the result. So the green signals represent areas with few low pH, well, the pH is fewer than 5.5, and red areas represent the size of osteoclasts. As you see, some red osteoclasts secrete acid, but others do not. And so bone destroying osteoclasts secrete acid in some limited, not all of the areas they covered. It seems that osteoclasts recognize the specific bone surface areas which needs to be destroyed. So yeah, in addition to osteoclast bone destruction, we could visualize many systems in bones, such as functional coupling between osteoclast and osteoblast, and also yes, or differentiation of osteoclasts in vivo. So and also the recently so I could visualize dynamic features of bone forming osteoblasts. Anyway, so I would say a bone is highly dynamic, and the Intravital imaging technique in hand is a very powerful tool for analyzing such dynamic phenomena. Okay, so next I will switch the topic and show our studies for identifying a novel macrophage population based on the imaging observation. So this macrophage is specifically responsible for pathological bone structure. And this was done by Hasegawa, a former graduate student in my laboratory, currently studying in Cambridge by a Human Frontier Science Program Fellowship. So the definition of osteoclasts are the cells having bone destroying capacity. However, it has been long controversial whether osteoclasts in inflammatory conditions, such as observed in arthritic joints, are the same or different from those in homeostatic or physiological osteoclasts. So actually, we can visualize both osteoclasts in vivo and found that the characters and efficiency for destroying bones are quite different. So normal osteoclasts, as I said, undergo transition between bone-destroying R-type osteoclasts and non-functional N-type osteoclasts, whereas pathological osteoclasts here are abnormally enlarged, they are very huge, and continuously destroy bones without any rest. Looks very different. And also, it's another imaging data representing bone-resorbing function itself. As you see, physiological osteoclasts seem to destroy only non-specific areas of the bone surface, as I said. Although pathological osteoclasts show a different behavior like this and destroy all the areas they covered. So these results indeed led us to wonder if these osteoclasts are really the same cell types or not. So in this study, we are trying to identify the osteoclast precursors in arthritic inflammatory joints. So the premise shows that homeostatic physiological osteoclasts in bone tissues are originated from CX3CR1 positive and LY60 negative, so-called circulating monocyte precursors. So as I said, basically osteoclasts are derived from a circulating monocyte. And among the monocyte population, we identify that CX3CR1 positive and LY60 negative monocyte or macrophage population can differentiate into physiological osteoclasts. So then how about the situation in an inflammatory condition? So what are the osteoclast precursors in arthritic joints? So to solve this question, so we collected blood, bone marrow, and synovium. Synovium is a specific tissue in arthritic joints. It's a pathological tissue. In synovium, bone structure apparently progressed. So we collected blood and synovial tissues, and then we analyzed the cellular population. So to make long story short, we found a new macrophage population only in synovium, which is a CX3CR1 positive or CX3CR1 high and LY60 intermediate positive fraction. So as I said, so this fraction is only present in inflamed joint tissues, but essentially absent in blood or other organs, including bone marrow. So what is this? Further analysis shows that this CX3CR1 high and LY60 intermediate population, we call region 3, is derived from CX3CR1 low, okay, CX3CR1 low and LY60 high, a so-called inflammatory macrophage, which can be called region 1 and region 2. So in summary, region 1 is an inflammatory macrophage in blood. And this macrophage can enter into synovial tissue. This is the pathological tissues in joint. And we rename these R2. And then R2 can convert into R3 population in situ. So, yeah, this is right. Anyway, so we focused on R3 because this cell type can only be found in synovial tissues and not in blood or in other tissues. So, as I said, the R1, R2, R3, we analyze submolecular expression by flow cytometry, and we found that this population, R3, this new population, highly express lung. So lung is an essentially molecules for osteoclast differentiation. So lung ligand is an important cytokine for osteoclast differentiation, and lung is a receptor for lung ligand. And as you see, lung ligand is highly expressed only in R3. Then R3 would be osteoclast precursors in this situation. And let me check. So concordantly with the high expression of lung, this population can preferentially differentiate into giant osteoclasts. So here we collect R1, R2, and R3, and then we cultured these cell types respectively with lung ligand. And then R3 can readily differentiate into lateral osteoclasts. R3 is more preferentially differentiated into mature osteoclasts. So therefore, we named these cells as arthritic osteogenic macrophages. We called it atom. So anyway, so based on the image data, we hypothesized that osteoclasts in pathological inflammatory joints would be different from those in physiological osteoclasts. And then we found that a new population responsible for pathological osteoclast differentiation, that was R3, or this is atom. So, and next we perform RNA sequences analysis for characterizing this new macrophage population, atom. So we collected R1, R2, R3, and then exhaustive RNA sequence analysis we performed. And based on the principal component analysis mapping, R1, R2, and R3 represent clear three distinct populations in terms of gene expression profile. So essentially we focused on R3, atom. Now we compare the gene expression of atom. Or we analyze gene expression of atom compared to R1 and R2, and red are the genes abundantly expressed only in atom. For example, these are integrin alpha 9, the ATP6.0D2. This is a very tough name, but it's a name of a kind of a subunit of proton pump. The cathepsin case is also important for the osteoclast function. Matrix metal protease 9, TRAP, integrin beta 3. All of these molecules are important for osteoclast function, for bone destruction. So that means R3 exhibits a highly osteogenic gene expression pattern. And we also found a master transcription factor for regulating gene expression in atom, which is a FOXM1. This is an upstream analysis of increased transcription factor. The FOXM1 would be upstream molecules for gene expression in atom. So, and we demonstrate that FOXM1 deficient condition, yeah. So allosteric bone destruction is clearly inhibited here, while bone homeostasis and skeletal development are completely normal in FOXM1 knockout mice. So suggesting that FOXM1 is important for generating bone destructive bad osteoclasts, but FOXM1 is dispensable for normal osteoclastogenesis for physiological bone remodeling. So based on the FOXM1 molecule, so we clearly distinguish physiological and pathological osteoclasts. To summary, so we identified a novel osteoclast for generating inflammatory osteoclasts named atom in allosteric joints, which is a 6 high and 6 intermediate and 20 high. The atom abundantly expressed lung and is ready to differentiate into osteoclasts, more differentiated than conventional osteoclast precursors. It's a different marker, 6 high and low. The atom can differentiate into osteoclasts without affiliation of N-FAT-C1. N-FAT-C1 is an important transfer factor for physiological osteoclasts. The atom rather depends on the FOXM1, right? But not N-FAT-C1. So master transfer factor is also different among inflammatory and homeostatic osteoclasts. So yeah, based on the interval imaging, we hypothesized normal and pathological osteoclasts may be essentially different. And now that this hypothesis turned out to be correct, we can separate good or bad osteoclasts respectively. And we found FOXM1 essentially important only for making bad osteoclasts. So by targeting such molecules like FOXM1, so we can only block bad osteoclasts without affecting good ones. And I think this will lead to a good therapeutic target for bone destruction in allosteric joints. Okay, so let's go to the last part. So for the last part, I so show the imaging of other immune system than the bones. So this was done by Dr. Kikuta. He's an associate professor in my laboratory. So besides bone tissues, we have established Intravital imaging system for analyzing cellular dynamic behaviors in vivo, such as skin, small intestine, lung, liver, other tissues, and so on. So also I have no time to explain all details, but now just show some of the interesting updates, such as lung. So here's an Intravital imaging of live tissues. And greens are macrophages, red are blood vessels, and many black, dark areas or alveolar spaces, because laser cannot pass through the air. And that clearly show the alveolar and blood capillary, and greens are macrophages. It's a control condition. So, and by means of this liver lung imaging system, so as a specific topic, we are trying to visualize event of interstitial pneumonia leading to pulmonary fibrosis, which is often complicated with rheumatic diseases. Interstitial pneumonia is currently common because it's also complicated with COVID-19. Anyway, so compared to the conventional pneumonia, interstitial pneumonia is so generally hard to be treated. So, and it was still unknown whether how we could treat these diseases. At the moment, so treatment against interstitial pneumonia or lung fibrosis are very limited. And so we definitely need a new therapy for treating these diseases. So we want to see what actually happens during the causes of interstitial pneumonia or leading to pulmonary fibrosis. So here we utilize bleomycin-induced fibrosis model. So as you know, bleomycin is commonly used for inducing a fibrosis in any tissues organs. Especially bleomycin can induce lung or pulmonary fibrosis by applying intratracheally. So, and this model is very clear, and inflammation occurs at first, and especially the first 1 week, massive inflammation can occur in lung tissues. And thereafter, fibrosis can progress slowly. And around 2 weeks ago, from the bleomycin inhalation, massive fibrosis can be seen. So, and this is imaging in control condition, as you see, red pulmonary capillary, and dark area represents alveoli, represents the macrophage. So please remember this, and this is an image at day 7, and when inflammation can be seen in lung, and you can see the dilatation of the blood vessels, and macrophages are actively moving. So this is a typical imaging for inflammation. And at this moment, as I said, fibrosis has not yet started. And next, let's see the timing of the onset of fibrosis. That is day 9. Okay, please remember these images. And next, I show the image in day 9. So, and this images in day 9. So as you see, here the image is completely different, and you see many large macrophages. Yeah, like here, large macrophages are sticking on the alveoli. And interestingly, so it is a day 9, and thereafter, so 2 days after this situation, what happens? So these large macrophages have gone 2 days more later, and you can see the increase in blue signals. Blue signals represent collagen fibers, right? Fibrosis. Right? So in summary, in bleomycin-induced lung fibrosis model, so we found a characteristic macrophage population only with the initiation of fibrotic change, right here. And then next, so we collect these large macrophages from bleomycin-induced mice, and then transfer into healthy mice, and found these macrophages can cause lung fibrosis by themselves. So therefore, we assume that this is a unique pathological macrophage directly inducing fibrosis. And now we are further characterizing these cell types. Especially, we could recently establish a method for isolating a large lung macrophage subset in fibrous condition. A large macrophage looks contain vacuole-like structure and possibly secreting several molecules inducing fibrosis. This is a single RNA-seq analysis of fibrotic lung. Red circle population correspond to large macrophages that we saw in Intervital imaging. And we are now searching molecules specifically expressed in this population, which would be a good therapeutic target against lung fibrosis. Okay, so and in addition, we also visual liver. And this is an example. And so because of time limitation, so I have to skip the details. But so we could detect a large macrophage also in the liver fibrotic conditions induced by NASH mode. NASH is a kind of so liver damage, commonly now common in so many countries. So and the most serious problem of NASH is a fibrosis in the liver. So and now we are trying to identify so specific macrophage having a capacity to induce liver fibrosis in this station. Okay, so yeah. So I think the advance in immunology, because I'm an immunologist, and I think that advance in immunology is fundamentally based on the taxonomic classification of different immune cells. And formerly, cells were classified by their morphology. And the great advances had brought in late 20th century. Cells were cells with identical morphology can also be analyzed by their cell surface markers by the development of monoclonal antibody, specifically recognizing the molecules and flow cytometry for classified cells stained by antibodies. So not simply analyzing cells, developing cell sorters enable us to specifically correct a certain cell subset and examine their specific function. So identification of a novel subset and clarification of their unique function have been the typical ways of research in modern immunology. And recently, another big advances have brought by comprehensive expression analysis at a single cellular level. And now that we can see exhaustive molecular expression pattern in each cell, we can classify cell population by the expression molecules, not restricted by cell surface markers. This single cell analysis technology has been revolutionized the various research field. So I have to say, a novel technology identifies novel subset. So however, even though these are indeed powerful tools for cell classification and identification, as a disadvantage, these techniques are destructive and missing their special temporal information. And we don't know the cell's location and also how they behave in vivo. And our laboratory is specifically interested in cellular dynamics in vivo and analyzing real cellular behaviors in vivo, mainly by using Intravital image technology that we have originally developed so far. And with this technique, we have originally we can visualize each cellular function at a single cell level and also classify the cells based on their dynamics. So recently, we have succeeded in correcting each living cell for comprehensive analysis and identify new cell subsets. So that, for instance, we have identified a novel osteoclast macrophage, as I said, and we also found so innate lymphoid cells in bone marrow responsible for bone marrow maintenance. We also subdivision lung liver fibrosis model and found novel types of macrophages specifically responsive for fibrotic chains in each organs. So I think these approaches are specifically useful for identifying novel kinds of macrophages because macrophages are plastic and easily change their phenotypes and characters in respective local environment, like chameleon. So therefore, we have to find them in their intact condition in situ. So they are missing when isolated in cell suspension. So, okay, so it's almost a last slide. As you know, all animals are moving, and precise control of moving or the movement is essential for maintenance of proper function. But they are moving as long as they are alive, and no movement cannot be observed in dead body. So therefore, I would say it is very important to observe inside animal in living station, and interval imaging is a powerful tool for analyzing the dynamics of life. So, yeah. And so, by the way, so based with a long-time close collaboration with a microscope company, Nikon is a world-leading microscope company. So microscope company. So Nikon Imaging Center, Osaka, was launched in Osaka University last year. And by means of this center, so we are actively collaborating all over the world. This is a picture in the opening ceremony last September. And in our center, several microscope setup, including multiphoton, confocal or super resolution, have been fully installed, and the talented special technical staff can support users at any time. So moreover, and one of the characteristic feature of Nikon Center is a commission service for Intravital multiphoton imaging. So as you imagine, Intravital imaging is not easy and needs several techniques and experience on microscope system as well as on animal experiment procedures. So, and our center can provide not only the cutting-edge microscopic system, but also provide original unique procedure for Intravital imaging from live animals. So this center is the first one in the world focusing on Intravital in vivo imaging. So also we are very welcome for the collaboration. Okay, so yeah, here I stop my talk. And here's a member. Yeah, so and especially acknowledge to Dr. Kikuta. Kikuta where? Here. So he's an associate professor of our laboratory, and he established an imaging system in many fields. Okay, so and yeah, these are public fund resources enable us to perform this study. Okay, thank you for attention.
Thank you very much, Professor Masaru Ishii. That was really impressive, that imaging. Seeing it really in a living mouse, cells migrating, so absolute stunning. For the audience, if you have any questions, please feel free to ask them. You have two options. You can either write your question into the chat box, or you can raise your hand and I will unmute you so that you can start asking your question yourself. And I see the first hand up. Thomas, you can talk now. It should work.
Hello, can you hear me?
Yeah, I can hear you.
Thank you very much for an impressive talk, Professor Ishii.
Thank you.
I have a question regarding macrophages. We were looking at lung metastasis and macrophages by intravital microscopy. And I was wondering, do you have an idea of how much plasticity there is between macrophage populations that you observe between one day and another?
I see. So now you're talking about the lung metastasis model, right? So I haven't examined the lung metastasis model, but I have experienced, or we have experienced imaging in lung, or so. I think so. Maybe as you know, in lung, there are at least two types of macrophages. One is alveolar macrophages, and the other is interstitial macrophages. And these macrophages are different origins. So alveolar macrophages are basically origin so from yolk sac, and interstitial macrophages originate from so bone marrow. So and in some conditions, for example, in your cases, so in so lung fibrosis or so intestinal pneumonia, so both subsets of the alveolar macrophages and also interstitial macrophages can differentiate or can convert into a different kind of macrophage that is different from those origins. So, and I think these macrophages are plastic in situ, and especially they're so changing their phenotype. So depending on there's some pathological condition, and I think also in the lung metastasis model, these macrophages can convert into some kind of pathological substance, I think.
Thank you.
Okay. Thank you very much, Thomas, for that question. We have another question in the chat box. I read that. It's from Jihan. Jihan is asking, how were the pH probe images analyzed? Which parameters, for example, spread of the signal or intensity? And which method did you use?
Okay, thank you for the question. So, so the detailed protocol have been already published in our paper. So please look at this paper. So, and briefly, so we quantify or basically, so we got images so based on the fluorescence of pH, pH probes, and we quantify the signals and compare to the background signals. And so by comparing the two signals by the background signals, we quantify so the pH, and also we can make some how can I say? Anyway, so we evaluate the pH signals by comparing the background, and yeah. So for decoding, we call signal intensity. But so as you see, the signal intensity can be changed. So depending on the imaging situation. So then we always got the background signals and compare. So and yeah. So but for the details, so if you are interested, so please look at our paper published in Nature Chemical Biology 2016, I think.
Great, thank you. I also have a question. It's a little bit maybe future perspective, but this intravital imaging, do you think in the near future or maybe it's already possible, and I don't know, do you think it will be also possible to even image subcellularly to look at organelles or something or the cytoskeleton?
I think it's possible, and at least for the case of the osteoclasts, that is, as you know, also the big cell, and for example, in mature osteoclasts, they have, for example, the 20 or 13 micron diameter, so it's very, very big. And at least a bit, so we can see the subcellular distribution of some kind of molecules inside the mature osteoclast in vivo.
Mm-hmm.
For example, we have several reporter system for conjugating, for example, some liposome or membrane protein. And so, for example, the proton pump is also there as the molecules expressed in the osteoclast, and we could see the subcellular distribution of a proton pump in the osteoclast. For example, we could see in some osteoclasts, proton pump can be distributed on the membrane, but in other osteoclasts, the proton pump seems to be distributed in cytoplasm. So, based on this experiment, we have already succeeded, and I think the spatial resolution is getting better and better, and in the future, I think in other cellular population or smaller cellular station, I think the subcellular distribution kind of some interest molecule can be visualized, I think.
Mm-hmm. Interesting. That's that would be really brilliant.
Mm-hmm.
There are no further questions from the participants. If so, you still have the chance to raise your hand or to type in. I might have myself one more question. As you have now identified this new type of osteoclast in arthritis, is it possible to transfer to apply that intravital imaging on human patients to really, maybe identify or understand better the disease and so on? Is that already done, or is it possible?
That's a very good question. So, so I haven't shown today, but so I'm also trying to make a new imaging probe, especially for usable for human or samples of patients. So because microscopy cannot be adopted to patients because now we're making some new instrument for direct attachment to the human skin or something to visualize the inside. So now we are making the instrument, and once we get the instrument, I think we can at least we can analyze, for example, the inflammation of the skin or some mucosal tissue, or because fluorescence cannot be passed through the deeper areas, so we can only see the near the surface areas. So at least we can see the skin or eye or mucosal tissue or something we can see. And for the case of bone traction, so we have to see the inside of the bone in human. So it is really, at least it's really hard. But in the case of the joint, we have so in clinical, so joint microscopy, endoscopy. You see, we have an endoscopy for seeing the joint. So, and if we can combine this system, we can see the osteoclast or bad osteoclast in human patient. I think that it's a relatively long future, but I hope it can be possible in long future, I think.
Mm-hmm. That would be really great and exciting. There's actually one more question from a participant in the chat box. I read that out. Thomas is asking a technical question. How do you deal with a TMR dextran taken up by macrophages over time? Does past labeling disturb future results?
Okay, so So this is okay. Yeah. So we often use the dextran. Yeah? So yeah, dextran or didabrodextran for visualizing blood vessels. Yeah? So we injected dextran, or first quantity dextran intravenously. Then we can see, we can visualize blood vessels. But so as he said, so dextran can captured by the macrophages, and so macrophages or red, or in this case, red signals can be accumulated in the resident macrophages. So does past labeling distort future result? Oh, maybe the question is is a uptake of or dextran or didabrodextran can alter the macro function? Was probably so. I'm sure at the moment I do not have the clear answers for the question, and yeah, probably so. These uptake may affect some function as a macrophage, but as far as know, so there, because the macrophages are continuously taking something, right, in your body. So not only the dextran, but also they are uptake several molecules. So, and I don't think uptake of dextran can significantly destroy or affect functional macrophages, because macrophages are continuously taking something. But we are not sure. And so for answering the question, so we have to check the function of macrophage. One is control, and the other is taking dextran, and then we should compare. But we haven't done such experiment.
Perfect. Thank you very much. These are all the questions. So thank you again for this excellent presentation, Professor Masaru Ishii, and for taking the time. It's really appreciated. And yeah, thank you again.
Thank you. My pleasure.
And thanks to everyone who attended.
Thank you.
Thank you.
Bye.
Okay, thank you, Jenny.
Thank you. Sorry for not introducing at the beginning.
That's right, not a problem.
Just simply forgot.
That's okay. All right, on to our next one. We'll work that out. Cheers.
In 1 hour, I see you again.
Yes.
Bye.
Thank you.